Hello everyone, welcome to my channel Online Biology Tutorials. In today's video, we will study gene regulation. We are already done with lac operon concept. Gene regulation means that how genes will be regulated, because translation does not happen every time. So genes are required to be turned off when protein synthesis is not required, and it has to be turned on when translation or protein synthesis happens. So first we will discuss in prokaryotes, and before understanding it, you must know few things like CAP. What is it? It is catabolite activator protein. I would request everyone to please watch lac operon concept first and then come to gene regulation, because it will get clear once you will watch that video. There is cap site as well. So cap site would be the one where this CAP or the catabolite activator protein will bind. Another molecule is cyclic AMP. So here C has to be written small. It is cyclic adenosine monophosphate. Okay. This is the secondary messenger actually, which will help in bringing some message. Okay, that's why I am writing here secondary messenger. Now these all components should be known to you, because then we will make the operon to make the things clear. Okay, you already know through last video what are the components of an operon. So it has a promoter for the regulator gene. This is our regulator gene. This is our promoter again. We have operator. Then we have structural genes in a sequence: LAC-Z, LAC-Y and LAC-A. This was the operon in the case of prokaryotes. It is seen in E. coli. Okay, so this is our operon. Now, if you talk about catabolite activator protein, it is present somewhat here, just a little upstream to the promoter. Okay, just a little upstream to the promoter we have cap site. Okay, this is our cap site, and obviously on this site what is going to get bind? Cap. Okay, but cap will not bind alone. It requires cyclic AMP as well. That means whenever catabolite activator protein will aggregate with cyclic AMP, then this complex structure will get bind to the cap site, and then transcription and translation will happen. So we will discuss two cases here by which it will be more clearer to you. The two cases are if glucose is present and lactose is also present. In lac operon, we have already studied that glucose is the most preferred source for bacteria to grow, because this is the most simple, which can be utilized easily by bacteria, right? So lactose will not be taken in case glucose is present. So how it is done? The glucose concentration in a cell actually defines the cyclic AMP concentration, and it is inversely proportional to cyclic AMP. Means if glucose is more, then cyclic AMP will be automatically less. However, if glucose is less, then cyclic AMP will be more. This is the main funda which you need to remember for the regulation of the gene in the case of prokaryotes. Okay, so in this example, if our glucose is present, here I have taken for you. So in case glucose is present, that means the level of cyclic AMP will be very, very low. But catabolite activator protein will be there. But when the cyclic AMP concentration would be very low or negligible, it will not bind to CAP, and ultimately the complex formation will not happen. If this complex is not formed, then cyclic AMP, this complex will not bind at the cap site. So what will happen ultimately? RNA polymerase will not bind, and if it will bind, it will not move on. That means the genes which are required for breaking down the lactose and to take the lactose in a cell, they will not be synthesized. So we have seen when glucose is there, Operon will be turned off. Means genes will not be synthesized because the cap site has not received the complex which will give a green signal to the RNA polymerase for doing the transcription. It will be halted. So that is why when glucose is there, Operon is always turned off. Right? Now we will see if glucose is not there and lactose is there, then what is going to happen? So again we will draw the operon, right? I am drawing just from here, promoter, operator, structural genes Z, Y and A, and the cap site will be located somewhere here. So this is our cap site. Now see here, what conditions are we giving? We are giving that glucose is not present, negative means not present, whereas the lactose is present. So in this case, gene regulation will be done like this way because glucose is not present. That means the cyclic AMP concentration will be very high. As I said, the glucose concentration actually defines the cyclic AMP concentration. So if glucose is not there, the level of cyclic AMP will be very high. This will bind to the cap site, and first of all, cap will bind to the cyclic AMP to form the complex, and this complex will bind to the cap site, right? So this complex is going to get bind at the cap site, which will allow RNA polymerase to bind with ACE, and this RNA will move downstream and it will help in transcription, and it will also help in translation. Okay, so once cap site receives the complex, RNA polymerase does its activity, and because of which the structural genes, they are translated. We already discussed that Z produces beta-galactosidase, Y produces permease, and A produces trans-acetylase, right? So this is how we have seen that if glucose will not be there, then Operon will be turned on. Okay, so genes will be turned on in terms of their requirement. They will not be turned on for lifelong. Okay, so this is what gene regulation is, which actually regulates the opening or the closing of the activity of the structural genes. Now we will see how gene regulation is done in eukaryotes. However, it is not there in the case of board examination, but you must know here because we have studied in prokaryotes, thus names can be learned by you. So eukaryotes also do have structural genes. They also have regulator genes like prokaryotes. They do have sensor genes which senses. They also have operator genes, and they also have enhancer genes which enhances the function, and silencer genes which silence the function. Okay, so enhancer and silencer genes are also present which regulate the gene expression. So we don't require to study the function of it, only the names are important. Gene regulation in the case of prokaryotes, it is important. I hope this was helping to you. If you haven't subscribed to my channel yet, please do so, and it will definitely be helping you because I will be covering whole book of NCERT. So you'll be getting all the tutorial videos at one place only. Thank you and stay tuned for the next video.